Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host. What if we could detect structural changes in DNA that traditional tests often miss? On this episode of DNA Today, we're diving into optical genome mapping, or OGM, as we'll call it, a cutting-edge technology transforming how we detect genetic variants and structural abnormalities. Our guest is Dr. Nikhil Sahajpal, Assistant Laboratory Director at Greenwood Genetics Center, GGC, who led the validation of OGM at GGC. We'll explore what optical genome mapping is and how it compares to existing genetic tests and what it means for patients and families worldwide. Dr. Sahajpal, thank you so much for coming on the show and exploring this really exciting technology with me.
Well, thank you, Kira, for having me here to talk about my favorite technology, optical genome mapping. Really excited to be here.
Favorite technology. All right, we're going to learn why that is the case. So, why don't we start by catching up listeners that may have heard of optical genome mapping but don't really know what it means? Maybe they've seen it at different conferences of like, oh, this talk is about this, but they didn't make it to that. So, can you give us an overview of what optical genome mapping is and how it works, kind of just at that high level before we dive into the details?
Sure. So, optical genome mapping is really a next generation cytogenomic technique. It gives a very comprehensive structure variation profiling of a genome. In principle, it is very similar to karyotyping. In cytogenetic circles, we often say that this is karyotyping on steroids. And that's basically speaking of the resolution of the technology that it provides. Procedurally, it is very simple. We isolate high molecular weight DNA, and the DNA can be isolated from any source material. For example, if we are talking about prenatal application, it can be amniocytes or chorionic villi samples. In the postnatal setting, it can be blood or skin fibroblast. In oncology applications, it can be bone marrow aspirates or solid tumor samples. Anything, any source material from which you can isolate high molecular weight DNA. So once the DNA is isolated, the DNA is labeled at a 6 base pair sequence motif. And this 6 base pair sequence motif actually repeats itself throughout the genome. For example, if you randomly sample any 100 kb unit in the genome, you will find 15 such sequence motifs or 15 such fluorophores now that we have labeled it, or we can simply refer to this as 15 such labels. Once the DNA is labeled, it is loaded onto a flow cell which sits inside an instrument where, with the help of electrophoresis, the DNA is linearized as it passes through microchannels and then through nanochannels. And at a single molecule resolution, this labeled DNA is captured by imaging. Once all the images are captured, these images are converted into a digital barcode, which is utilized to make a consensus genome assembly. So a de novo genome assembly is assembled without any reference here, just using the patterns of these labels. Once the entire genome assembly is built, it is then mapped to a reference genome, any reference genome it can be. And because we know the position of this 6 base pair sequence motif, we know the label pattern of the reference genome, and comparing the genome of interest to the reference genome and the variation in these patterns, we can call the different classes of structural variations. So this is basically karyotyping, but with a much higher resolution.
It's really interesting because I come from a cytogenetics background. That's what my undergrad was in. So I've done so many karyotypes. And this is just the first time I heard about this. I was just so fascinated because I was like, wow, this is like a super, super high resolution karyotype in some ways. And it really is like combining the strengths of not just karyotype, but multiple cytogenetic technologies. I'm also thinking about microarray and FISH. Is that an accurate statement to make, that it's sort of combining like karyotype, microarray, and FISH together?
Absolutely, that's very accurate. And because you are a cytogeneticist, you picked it up. And if we talk about each of these technologies, right, karyotyping, chromosomal microarray, and FISH, what we know is that karyotyping is actually very accurate for genome-wide structure variation analysis, but there are limitations we know of, that the resolution is not that great. We can look down to 5 MB in certain cases, 10 MB, so to say. If we look at chromosomal microarrays, ideal for copy number variation detection. But then if we talk about duplications, even within the copy number variation, we do not know the orientation of those duplications. Are they in tandem? Are they inverted? Are they sitting just next to the duplicated material, or are they inserted elsewhere in the genome? Not even talking about balanced rearrangements with chromosomal microarray, but just the copy number variation detection is also limited, not knowing the positional or the orientation of the duplicated material here.
Because you don't have a map like a karyotype. You can literally see what's
Absolutely.
happening. With a microarray, you're just like, okay, the like bits are there or not, but you have no sense of where they are on the
Absolutely.
chromosomes or genome.
Absolutely. So having that structural overview of looking actually at the long DNA molecules is very parallel to what we do in karyotyping. So you can actually see it. And this is like precisely what cytogeneticists miss, seeing the structure, seeing the chromosome, seeing the DNA, how it really is positionally and orientationally with all these rearrangements.
I want to ask a controversial question because as I said, I passed the certification. So even though I don't do karyotypes anymore, I could. Is that going to be an outdated skill? Is OGM going to replace karyotypes?
So there are multiple layers to this question. Simply speaking, I would say in several of the circumstances, it does replace karyotyping just because of the resolution and what it can detect. And I'll say here that there are, with each technology, there are advantages and there are disadvantages. There are few blind spots. So optical genome mapping does have blind spots. If we compare it to karyotyping, the blind spot here would be that it cannot detect Robertsonian translocations. So the other limitation would be that it does not pick balanced translocation if the breakpoints are in the centromere, just because we do not know the sequence context of the centromeres. So there's always utility of multiple technologies. But if we are talking about what should be our first year test, I would say that it has to be optical genome mapping now, where we are with respect to the ease of doing the assay and the resolution and the information that it gives us, that karyotyping cannot be at this time point be our first tier test to go for. So
That makes
yeah,
sense.
and there's different layers to it in constitutional setting, prenatal setting, in oncology, which needs a much wider discussion. But if the simple question is, does it replace, then yes, it does replace karyotyping.
Yeah, which is it makes me a little bit sad as someone that got really good at karyotyping years ago. But at the same time, we got to go with the times here. But I'm happy to hear that there probably, hopefully, for me, of just loving my chromes, that there will be a time and place for karyotypes. And I think Robertsonian translocation is a great example of that. And we have that in prenatal and in cancer. So that is relevant. I can tell that you're so passionate about this. And you said at the beginning that this is your favorite genetic technology. I'm curious first how you personally got interested in optical genome mapping.
Yeah, so if you see, like, if we reference the evolution on the molecular side with sequencing, what we see is that we were relying on Sanger sequencing, Pyre sequencing, very like single gene sequencing. And then we moved to next generation sequencing from panels to exomes, now to genomes. But this evolution was not so much so on the cytogenomic side. So we had karyotyping, we had fish, we had evolved to chromosomal microarrays, each having their limitations, but it was not keeping the pace with the molecular side or the sequencing side. So what we have now with optical genome mapping, and why I said that it's one of my favorite technologies, is because precisely this, that it is the next generation level in the cytogenomic domain. So it's really a jump in that domain. So that's the fascination with the technology, that it really not only replaces karyotyping, but it replaces chromosomal microarray and fish, and it combines the advantages of all three technologies with a much higher resolution. So just to speak on the resolution, it is actually 1000x fold higher resolution than karyotyping, which is which is huge enough, right? And you can see those balanced rearrangements, translocations, inversions, and just like swapping of different materials, which was, we would say these events are cryptic to what we have right now.
So then how about Greenwood Genetics Center? How did they first get involved and then bringing you on as an expert in this area?
If you're following the Greenwood Genetics Center story, it is this that there's a culture of innovation and there's a culture of looking into technologies, techniques, and methods that can help better serve the patient population that we cater to. So in this culture or in this scheme, we as a center looked at this technology. And when I joined Greenwood Genetics Center, I was working before joining the center here at a medical college where I was using this technology in oncology applications. So I came with some of the experience here, which helped now bring this technology as a clinical test and we offer to our patients.
So we've talked about how impressive this technology is and just how we're able to look at so much more and almost combining a lot of technologies into one with OGM. I'm curious, how is GGC using this test? What other conditions or patient groups do you see as strong candidates for OGM testing? I want to hear like how we're actually using this.
Yeah, so we at the genetic center, our main patient population is rare disease population. So we offer optical genome mapping for the postnatal rare disease application as of now. We offer optical genome mapping in two different workflows, so to say. One is optical genome mapping targeted, and one is optical genome mapping complete. So if I briefly walk you through both of these workflows, it is this that in optical genome mapping targeted, physicians or providers can actually ask us to look at precise regions of the genome. These can be where, for example, they had found a balanced translocation with karyotyping, and they want to map the breakpoints to see if it disrupts a gene, what's the context of the balanced translocation, is it really balanced or not? Or they can ask us to look at a specific gene where they might have found a variant with sequencing technology and the inheritance pattern here is recessive mode of inheritance. So we are looking for a second hit, or simply there's a strong phenotype genotype correlation and there might be a structure variation which is missed with the prior testing, any of the balanced rearrangements, inversions, or repeat expansions or contractions. So precise narrowed down regions to be interrogated will fall under optical genome mapping targeted. Then there's a test which is optical genome mapping complete, which is similar to any other test where we would look at genome-wide structure variation interrogation of the genome and will compare the structure variation and the genic context to the phenotype and see if the structure variation would explain the phenotype that we are seeing. Beyond this, we are working to bring this on for other applications as well.
Yeah, it's exciting just to see how much we could do with this technology and how we can really impact people's lives. Of everything that we do in genetics goes back to how can we help people and patients. I was learning about a recent study that you did about neural tube defects. So for people that are like, oh, that sounds familiar, that's where there's an opening in the spinal cord that can cause different problems, often mobility issues. So OGM identified variants in about a third of cases, like 30% of cases, that other methods missed. So being able to identify that, which that to me is a very high number. I would love for you to talk about the discovery of these novel candidate genes and what their potential role could be in neural tube defects, especially as I mentioned I've worked in cytogenetics, but I practice as a prenatal genetic counselor. So I'm really intrigued to hear what you have to say about this and just talking about this study.
Yeah, absolutely. So, neural tube defects have been a really challenging cohort for the past several years. This might be the only condition actually where we know what prevents it, like folic acid supplementation and fortification, but we really do not understand the causation of these defects. So it's
I've never
very,
thought of it that way. That's such a good
very
point.
unique with respect to rare conditions. Greenwood Genetics Center has been involved for a long time in prevention and identification of causation in these defects. And in this, as part of this effort, we utilize optical genome mapping to identify genetic causes in these birth defects. And what we know from studies done all over the world is that if you do comprehensive testing with whatever available techniques we have right now, the diagnostic yield is actually 8%. So there's 92% that we do not understand at all. So seeing 30% of candidate findings in our cohort is very encouraging. And speaking to the resolution and what this technology offers is actually looking at the blind regions of the genome. So a lot of these findings were actually events where the breakpoints are in repeat regions of the genome.
Because that's really dense and it's hard to look through when it's like the same letters over and over.
Absolutely. So the way we can see it is actually by karyotyping, but then it needs to be big enough. And the problem with short read genome sequencing is you need reads which can span these long repeat regions that you can get unique sequence context. So having long DNA molecules with this technology gives us that DNA sequence motif to actually span through these unique regions, repeat regions, and really capture these structural variations. So we are really excited about what we saw with these defects.
I find it interesting and helpful to hear how is this actually going? Because it's exciting to talk about what are these aspects, right? We talked about the high resolution and just what we're able to find that we can't in with other, especially cytogenetic technologies. It's nice to see it illustrated and like as an actual example. Are there any early success stories that come to mind where optical genome mapping did provide a diagnosis where these other methods failed?
Yes, absolutely. So we have several of these, and the end goal here is actually to provide answers to patients and families, and that's what we do here. So just to share a couple of cases here, we had a family where there were 7 affected individuals in 3 different generations. This is how genetic diseases often run in families. And we had done a battery of tests from karyotyping, chromosomal microarrays to whole exome sequencing, and then other panels, and all were unremarkable. So once we brought optical genome mapping as a clinical test, we offered this to the patient and the family, and we actually found a very balanced event on chromosome 7. And the event is actually such that it does not fall under any of the structural variation definitions like deletion, duplications, inversions. So it does not fall under any of these defined terminologies of these structural variations. And what it really is actually a swapping of material. So simply switching of 2 genomic regions which are sitting next to each other. So they swap position. And just because they swap the position, it included the gene DLX5, which is associated with autosomal dominant split hand, split foot malformations, which explained the phenotype in this family, and it segregated in all the affected individuals. So it is just this positional information, and the causal mechanism here is that the gene moved away from its enhancers. So since enhancers control the expression of the gene, they being moved apart, the enhancers could not regulate the regulatory control over that gene, and the gene could not express in specific tissues. So
So it's kind of like the on-off switch wasn't right next to it. So the gene couldn't be expressed in the right amount.
absolutely.
And so just the position of the gene, which because it sounds like it was so small, you needed that high resolution to find it. Like all the other
Absolutely.
methods failed. It was OGM that was able to identify such a minute change.
This is precisely correct. So again, like if this event was 5 MB of segment moving, we would have seen it with karyotyping. But because it is only few KBs, you cannot really see it. And here's again going back to that cytogeneticist obsession with looking at the chromosome or DNA, because you can precisely see what is happening. So having that context here is really important.
And that's a good case, I would think, that's to look at optical genome mapping because you're saying it's 3 generations, it seemed to be autosomal dominant. You're like, there is some kind of genetic change that's leading to this phenotype, these symptoms, but we just haven't found it, but there certainly is one. So that makes sense as a case to be an earlier one to show, hey, this is how this technology can find this.
Absolutely.
I think you were thinking of another case too.
Yeah, so this one is actually alluded this fact earlier, dual diagnosis and reducing the diagnostic odyssey. And what this case is, so we had an individual which presented with Down syndrome picture and was actually diagnosed with Down syndrome on day one of his life. Karyotyping was performed and we saw trisomy 21. But then by age 7, started having difficulties in walking, which is not a typical feature of Down syndrome. And this difficulty in walking progressed, and by age 11, the individual was non-ambulatory. So it really progressed. Again, all the possible testing was done, from panels to exome sequencing to chromosomal microarray to karyotyping to SCA panels, repeat panels. And because there was a suspected muscular dystrophy, there was nerve biopsy, muscle biopsy done, everything was unremarkable. And then we performed optical genome mapping when this individual was age 15, and we found a biallelic expansion in the gene FXN, which is consistent with the diagnosis of Friedreich ataxia. So at age 15, actually, we got the second diagnosis for this individual. But thinking of it now, since we were talking about would it replace karyotyping or not, if optical genome mapping was performed on day one, the individual would have had a dual diagnosis on day one of his life. So would have had Down syndrome, would have had a diagnosis of Friedreich ataxia, and the family would have been prepared for what is to come rather than chasing the second diagnosis here. And just to share another note on this case and family is that just when this diagnosis of Friedreich ataxia was made, the first drug for the treatment of Friedreich ataxia was approved by FDA. So now this individual is actually on the treatment for Friedreich ataxia, and we are rooting for this patient and the family. So these are the kind of cases and stories which actually motivate us and is the reason that why we are doing what we are doing here.
And that's such a great example too of just because you have a diagnosis doesn't mean there's not a second diagnosis there, where if you do have a diagnosis, it may not explain everything. And then you have to say, okay, well, we can't just stop there. There could be another condition. And that I feel like isn't talked about a lot in genetics.
Absolutely, it is
It's
not. It is not. Yeah, and that's the whole reason for advocating for comprehensive genomic profiling is precisely this: making the diagnosis, making dual diagnosis where it is to be made, and then all the other applications that can be done with comprehensive genomic profiling.
Yeah, earlier in the year, episode 355, we had an interesting episode. A good chunk of it, we were talking about dual diagnoses. And yeah, this is just a laundry list I feel like we've made during this episode of reasons that optical genome mapping should be a first tier test. Other types of testing should maybe then be second tier, like a karyotype, or if there's a very specific need. Let's say I have a patient that has a Robertsonian translocation, they do an amniocentesis, and they're looking to see, did baby inherit this? That would be a reason to just straight up do a karyotype. But yeah, it seems like this is where the future is headed, to be really starting with optical genome mapping. Any final thoughts before I let you get back to working on this really exciting technology?
Yeah, I would say so. So using it as a first year test is an ongoing discussion, and then there are multiple layers to it. What I would say is that labs that are using chromosomal microarray and whole exome sequencing as their first year test, this is where optical genome mapping can actually replace chromosomal microarray, and it should be chromosomal microarray. Instead of chromosomal microarray, it should be optical genome mapping and whole exome sequencing. For labs that are actually doing whole genome sequencing as a first year test, I think that makes sense that whole genome sequencing should be the first year test. But because the diagnostic yield there would be anywhere between 30 to 50%. It should be that optical genome mapping should be used in the whole genome sequencing negative cases as a reflex test. So there are multiple layers to how different tests are used. Several of these circumstances, optical genome mapping would be the first year test. In many of these circumstances, optical genome mapping would be a combinatorial supplementary test. So based on the application and what is being used, there are different uses of this technology, which would fit well with the workflow.
Yeah, I think that's an important point, that we don't want to cast aside whole genome sequencing, because in many, many cases, that should be the first tier test. Just like so much, it depends on the situation. And, there's so many exceptions and there's rules in genetics, but then it's like, oh, but in this case, we do that. And that's why we're both employed, right? Because there's just so much to learn and figure out. Dr. Sahajpal, thank you so much for coming on and nerding out with me about this technology. I always love going back to my cytogenetic roots when I have an opportunity. And then we even got a little prenatal spin in there at some point. So I really enjoy just learning about where we're headed with this technology. And for those that are going to be at the big genetic conferences coming up in a couple weeks and later this fall, ASHG in Boston, NSGC in Seattle. I'm going to be at both of those. Can't miss it. I feel like they're big social events for me as well as learning. And Greenwood, I have to say, you guys have pretty awesome swag. I wear my my black cap all the time. You see me flying to these conferences, I'm often wearing it. So definitely stop by, chat with Caroline there. And she knows so much about this. So certainly stop by the booth there, because I will probably be hanging out there as well.
Well, thank you for having us. It was lovely chatting with you, and we'll be at ASHG and NSGC. Looking forward to it. Thank you.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.